All right, everybody. So let's welcome Pramod. He will now, travel with us to Mahabaleshwar, and I'm quite happy that he managed some time to come after a lot of negotiations. So thank you, Pramod. And he will talk about something which is quite different from what you have been hearing so far. But somehow, life is all networked and linked. So I think this is also will be linked to axonal transport sometime.
Thank you. Okay, so I'll be talking a little bit about mechanics in relation to axonal mechanics. So I will. So this is where I come from. It's Raman Research Institute. That's the main building of the institute, and on the right side is the founding father. It's Bangalore, and this is going to be the plan of my talk.
There is a pointer on the roster.
There's somebody's cell phone also, it's ringing. Sorry?
Can you please go and find out the later point?
Somebody's phone is ringing here. So I don't know. It's maybe room or you. Pointer is supposed to be here. Even a stick will do. There was a pointer. Okay, fine. Yeah, there's no stick, is it?
Anyone seen the later pointer?
Okay, anyway, I'll start. So,
we will locate it.
So, I'll give a brief introduction to what is called viscoelasticity. I guess it's something which will be new to most of you. So, I'll try to give a very simple introduction to this particular material property. Then we will discuss why understanding viscoelasticity is important. It can also be important if you are trying to look at mic. Okay. Is that better? So we will discuss why understanding viscoelasticity is important, also for transport. We'll discuss a little bit of axonal mechanics, both active and passive, and I'll also discuss some experimental methods which are used to study axonal mechanics, and then maybe in the end some take-home messages and outlook. Okay. So let me start with by introducing viscoelasticity. So you are all familiar with simple elastic materials like a spring, for example. So if I take a spring and if I stretch it, these are called Hookean materials. Then I can write down the force as, so x is the distance by which I am stretching the spring, and k is the spring constant. If I take a stiff spring, the k is larger. If I take a soft spring, k is less, and so on. So if I take the spring and if I apply a step force, this is time. Okay, so I suddenly apply a force on the spring, then the spring will stretch instantaneously. Okay. It will stretch instantaneously. Now, if you take a liquid, so these are the solids which you're familiar with, like for example a crystal or something like that. And the other type of material you're familiar with are liquids. And if you take a liquid, then let's say that you have a sphere which is moving in a liquid under some force. Okay. So this maybe Gautam discussed a little bit on viscous drag and so on. So you know that if you have a liquid which is moving like that, then there is a force acting on this liquid, which is given by. So this force now depends on the velocity, how fast this sphere is moved. Okay, or if I have a constant force, then I get a velocity which depends on the force which I am applying. So if I do the same thing as here now, this is force. I suddenly apply a force. Okay, then I get a response which is again x. Okay, at some point it will reach terminal velocity and it will move with a constant velocity. Okay, so these are the materials you are more familiar with, and in terms of stress, I can write, if I want to calculate the force, here I can for a bulk material I can write stress as, okay, epsilon is the strain of the material, sigma is the stress, and the constants in front is the Young's modulus. Okay. Similarly here, this would read as eta is a viscosity. Okay. Here the force which it experiences depends on the velocity. Okay, the faster the particle is moving, the greater is the viscous drag. So here the stress depends on the strain rate. Okay, not on strain. So now what are viscoelastic materials? So viscoelastic materials, I can give you a okay. So here, here I draw a spring to represent a elastic material. So for the viscous material, I can draw what is called a dashpot. Okay. A dashpot is nothing much. For example, if you take a syringe and fill it with some viscous fluid, and if you apply a constant force, then the fluid comes out at some rate. So it limits how fast I can push at what rate the piston is being moved, right? So that's why it's usually represented by a cylinder and a piston. So it's a purely viscous element. Now a viscoelastic element can be constructed in many ways. A simple case is this. Okay. So I am putting a spring and a dashpot in parallel. Okay. When I do that, and if I apply now, if I try to deform this material, you can see that the strain for this and the strain for this is the same because they are connected in parallel.
One we see the doors in the lecture hall.
Sorry?
The doors in the lecture hall.
Yeah, I'll come to that. Exactly. So I'll come to that. So yeah, thanks. Okay, so here that epsilon is a strain because they are connected together in parallel. Okay, so epsilon, if you like, is the displacement which you are applying or the strain which you are imposing, and the stress is, okay, because these two things add up. Okay, they both contribute to stress. And we know that from what I wrote above, this can be written as, okay. So sometimes I'm interchanging epsilon and x. This basically is a strain. Okay. So as Gautam pointed out, a good example of this are the door closers which you have. Okay, so if you want an automatic door closer, the simplest thing would be to just attach a spring. Okay, but if you attach a spring, then if somebody opens the door and leaves the door, it's going to bang shut immediately, instantaneously. Okay, because the spring has this sort of a response. But so to prevent that, what they do is they also add a viscous element to it. Okay, like something like a syringe and a piston. Okay? So that the spring does not, the door does not bang shut. It shuts with a characteristic time. Okay? And it's the same thing which I used in shock absorbers in vehicles, for example. So now if I will not go into solving these equations. You can solve these equations and you can ask what happens under various conditions. Again, as I did there, I will pictorially tell you what will happen. So if I now take a material like this, and if I do that, all of a sudden I apply a force to it, you can see that it cannot stretch instantaneously because this is a viscous element. It takes time to respond. Okay, this cannot respond instantaneously, whereas the spring can respond instantaneously. So the net effect which you see is a response like that. Okay, there is a constant force acting here. Okay, the spring cannot extend instantaneously, but at some long time, if this dashpot can relax, then the force which I apply can be exactly balanced by the spring. So there is mechanical balance. So this is a state where you have reached mechanical balance. At this state, this material has gone from this to this to something like that. Okay, it has stretched by some amount. Okay, and the force is now balanced entirely by this spring. So this height gives you a measure of the spring constant of the spring, and this time taken, the relaxation time, what's called relaxation time, gives you a measure of the viscosity. Okay, so there is a relation as this goes as this curve. Okay. And tau is a relaxation time. Okay. So if I have a material like this, if I apply a step force, then I can measure its response, and then from this response I can calculate what is the spring constant and what is the viscosity. So many material like rubber, for example, and cross-linked polymer network and tissue, they all, under very small deformations, they all behave roughly like this. Okay? We'll get into a little bit more detail later. So you can think of other type of material. For example, I can now connect the spring and the dashpot in series. I can again write down equations similar to the earlier case. In this case, the strain adds up. So D stands for dashpot and S for spring. And I can again ask, how will this material respond? Okay. So again, you can solve these equations to calculate the exact response. I'm going to give the solution picture here again. So I am again doing the same test. I am applying step force to the material. And now you can see that again, the viscous element cannot respond instantaneously. The spring is going to respond instantaneously. So this element can stretch immediately. Okay, because the two are in series. So this element will first stretch instantaneously, and then what will happen? There is a viscous element here. Okay, so there is no equilibrium, and this thing can keep stretching forever. Okay, so after the initial stretch, it will go to this. Okay, and this initial stretch will give you a measure of the spring constant, and the slope will give you a measure of the viscosity. Okay, because at long times it is only the dashpot which is contributing. And this sort of response, so the difference between that case and this case is that there, if I apply a constant force, irrespective of how big, small or big the force is, there is an equilibrium. Okay, there is an equilibrium strain or equilibrium length. Okay, the system does not keep extending forever. But in the second case, that is not what is happening. I apply a small force, it can be as small a force as I like, but the system will keep extending forever. Okay. The more I weigh, the more the strain becomes. So that class of material are called viscoelastic solids, and this type of materials are called viscoelastic liquids. Okay. Sorry?
The elastic part of
Yeah.
the response, is it going to be exactly, if you have the same spring,
Yeah.
is it going to be any different?
The elastic, there is going to be a difference because here finally you have some, depending on the force you are applying, there is a drag force coming from the, there is a drag coming from the dashboard.
Rather, if we do these two to scale,
Yeah.
what you have drawn here,
Yeah.
let's assume the same spring,
Yeah.
what would they look like?
So this height will be same as that equilibrium strain.
It will be the same.
It will be the same. Okay? Because at t equals 0, all the force is exactly balanced by this spring alone.
But this, will it be really that sharp when it goes up? Yeah.
So this can respond instantaneously, right? So at t equal to 0, this will give me an extension which is solely coming from the spring.
So, but there is a viscous drag also, so it is not only
Later, so as soon as I pull, the instantaneous response is coming entirely from the spring.
So that's what I'm asking. Will the response of the spring be complete before the viscosity starts taking effect? Is what I'm wanting to.
It will be. Okay, and there will be a region here where the viscosity is taking over. So if I take a t equals 0, if I ask what is the extension, then that gives me the spring constant. And in the other case, I go to t equals to infinity, or t equals very larger than the relaxation time, and then I ask what is the extension, that gives me the spring constant.
Even if you are in a viscoelastic medium,
Yeah.
you can very precisely measure the elastic component without interference from the viscous component.
If your system can be modeled in this way or that way. Okay. Most real materials are more complicated than that.
how does it happen that even with a small force it will go on extending?
Because this is like I said, this is like a syringe and a piston, right? If I take a syringe, fill it with water, and if I press on the, if the friction due to the from the piston is not too high, if it's just a liquid which is contributing, irrespective of how small a force I am applying, the piston will keep moving. Right? It will move at if the force is constant, it will move with a constant velocity. So that is the reason why here you get this slope. So what kind of material shows this? This can be seen in polymer solutions, and polymer solutions are a little bit more complicated than that, but they are viscoelastic liquids. And I have a sample material here which I'll pass around. If you take this is some kind of a putty which I bought from a toy shop. So if I take this, if I roll it into a ball, let me just, okay. And if I throw it, it bounces. Okay, so you will say this is an elastic object. It bounces very nicely. Okay, but if I just leave it on the table, if I keep it here for, it will take long time, and if it come after 1 hour or something, it would have flown down. Okay, it behaves like a liquid. So what is happening with this? I can pass this around. So like in this simple case, for example, whether it behaves like a liquid or whether it behaves like a solid depends on the time scale. Okay, at very short time, viscoelastic materials can behave like a solid material, and at long time it can show viscous properties. Okay?
Like us.
Like us. Yeah, exactly.
For a long time you will be.
Yeah. So. Let me see. Okay, is that clear? So I can ask a question. For example, I can ask if I take Mount Everest, is Mount Everest a solid or a liquid? What do you think? Okay, I fix temperature and pressure. I don't change temperature and pressure. How many of you think it's a solid and how many of you think it's liquid? Solid. Solid. So you think it's liquid. Okay. So the correct answer is it depends on the time scale. Okay. If I ask is Mount Everest a solid in a time scale of a day or a few days or a month, then it's a solid. Okay, because if I apply a force and if I see what happens here, it doesn't move. Okay, but we know that, as he said, it's growing. Okay, it's growing due to the pressure from the two plates which are moving against each other. Okay, because the subcontinent is pushing against the rest of Asia. So at long time scale, it's like a liquid. It's deforming. It's deforming and it's increasing in height. Okay, so it depends on time scale. Okay,
so actually, I know glass is also considered over long periods of time. So how do we know the objects which we day-to-day life? How many?
Okay, so one way of knowing this, it's a good question. So one way of knowing this, one test are these things, what I already mentioned. Okay, you can also do. If I have a solid material, often I cannot deform it by very large amount. Okay, and I don't want to deform it by a very large amount because I want to be within some what is called a linear regime. Okay, if I deform it by a very large amount, other things happen. It can yield, it can flow, and other complications. So I plot stress versus strain. If I have a Hookean material, then this will be a straight line. Okay, and if I cycle it, if I do my force, I'm cycling like that, then I keep going back and forth along this line. Okay, but if I have viscoelastic material or viscoelastic solid, then there is dissipation. So then in that case, this will not be a straight line. This will always be a loop. Okay, this will have the curve. When I stretch the material and when I bring it back, the curve will not retrace itself. Okay, because of the relaxation which is happening in the material and dissipation which is happening.
Okay. Isn't this also seen in Hookean material? This is hysteresis, right?
If you see hysteresis, it's not an ideal spring. Okay, or it's not an ideal elastic material. There is dissipation. In fact, any material you take, if you take water, again, like I said about Mount Everest, water can behave like a solid at very short times. But at long times, it's a liquid. So, any material you take, if you look very carefully, you will see that there is some dissipation, any real material. Okay, so the ideal elasticity is in most cases an idealization. So the area under this curve gives you the energy which is dissipated, okay, in each loop. So this is one way to test whether it's an ideal elastic material or a viscoelastic material. And then the other things are like these tests which I mentioned.
In the physical sense, as if like if you look at materials, how do you know whether it is in parallel or series?
You don't know. What you do is I give you a material, it's like a black box to me, like ropes with black box. So, and then what you do, all I can do is I can do some tests on it. Okay, I'll show you examples. Okay, so I can pull on it and I ask how is this material going to respond to the force I am applying. Right? From that I can tell, from that response I can tell which sort of material it is likely to be.
And if you are making such a material?
If you are making such a material, of course people want to make such material. For example, if you are designing a car, I want to design a shock absorber for the car and I want to dissipate the energy. If the car goes through a pothole, then that energy is to be dissipated as quickly as possible. So then I need to calculate what should be the ideal viscosity and ideal the spring constant. Okay, then you do modeling. So just as Rube was mentioning about modeling for molecular motors, engineers do develop models where they adjust these parameters and see, they put in the weight of the car and various things and they optimize. Okay? Or a door closer for that matter. Okay, so maybe I can leave this here because mostly I will now. So no trace of the pointer, is it okay?
Pointer is probably somewhere on that area.
Okay. I'll manage it.
No, okay.
Oh, you flip it.
No, I think.
No.
After that, must have gone below something.
Here, right here.
Oh,
You kept
okay.
the papers on
Thank
that.
you. Thanks. Okay. So I'll skip all this because you have heard this many times. Okay, so I come to one of the early experiments which was done in 1984. This is not the first experiment, but one of the nicest experiments where I also Another reason I want to show this is also because this is cool on transport. Okay, so what these people have done is that they put a magnetic bead inside an axon. Okay, it's a squid axon, so the diameter is about 500 microns, if I remember correctly.
Sometimes a millimeter.
Sorry?
Millimeter.
Or yeah, almost a millimeter. This bead is about 100 micrometer, so it's a fairly large bead. And you inject this bead inside or push it in from one end. You cut the axon, push it into the axon from one end, and this bead is trapped in the cytoskeleton. Okay, the axonal cytoskeleton. And now what you do is you come with an electromagnet. Okay, this is the tip of the electromagnet, and you can pass a current through the coil, and you can adjust the magnetic field. So when I switch on the switch here, when I switch on the switch, there is a current going through the coil, and this gets magnetized. Okay, when it gets magnetized, because it's a paramagnetic bead, there is a force acting on the bead. Okay, and if you do it carefully, you can have a condition where the force is almost axial to the axon. Okay? And then they look at the response of the bead. And what they see is the following. What I've shown in green is the force. So they switch on the magnetic field at this point, time equals 0. Field is on, so the force is here, the constant. And at some time you switch off. Okay? Then the force goes to 0. And what you measure is the position of the bead. Okay, and you see that the position of the bead, if you had done this experiment in liquid, you would have seen that the bead moves with a constant velocity. Okay, because this is nothing but the drag, like the Stokean drag. Okay, so it will move with the terminal velocity. But that's not what you see here. You see that there is a characteristic response. There is a initial increase in strain, and then there is a constant slope here. Okay. So from this experiment, again from considerations like what I described earlier, they conclude that this material is made of a combination of springs and dashpot like that. It can be not made of. This can be modeled by this sort of a system. Okay.
Why not only viscous drag is responsible?
If it is only viscous drag, I rubbed it off earlier. If it is only viscous drag, you will not see this. Okay? You will see only this part. Okay. Yeah. So if you really calculate these things, you will see that there is one term here where only the viscosity is coming in. Okay, and this is proportional to time. Okay, at long time, this is the only term which is surviving. Okay, and this is coming purely from the viscous drag. So viscous drag comes at very long times. Okay, at short time, it's a combination of this. Okay? So I can just qualitatively explain these equations. At a very short time, as soon as I apply the force, this spring is going to stretch instantaneously. Okay, so there is a jump here. You can see there is a jump. Subsequently, this system is going to relax, okay, exponentially, like I mentioned earlier. So this gives rise to an exponential relaxation, and this element, which is in series, gives rise to a continuous strain. Okay, so continuous straining of the material, and the combination of that is giving you this part of the curve and this. Okay. And you can actually, what these people have done is to fit this, fit the strain which they calculate out of this, and you can see that the continuous line is the fit. It fits nicely. Okay. And what happens if I switch off the magnet? Here you're switching off the magnet. Because of this, the material has already moved by some amount in a non-elastic manner. But there is always some elastic energy stored in the spring because of the viscous drag. So any moment when you switch off, it will recoil. Right? Is that clear or not? It will go back by some amount, and then it will settle at a new position, which is a force-free position. Okay? Is that part clear? Because this is also important. For example, if you are looking at some object which is getting transported in a cell, or not in the case of cytoplasm like what you do, but if it's in the case of axon, it's a very crowded environment. Okay? It's full of cytoskeletal filaments, and the vesicle, synaptic vesicle, for example, which is moving, has to push through this cytoskeletal network. Okay, so there is viscoelastic forces acting on it. So if I look at a particle, for example, if this particle is being pushed by a motor, a single motor, then this particle will move like that over a long time, and then if the motor switches off or the motor comes off, unbinds, then it will relax in this fashion. Okay, which means the particle will move back.
Have they tried this with different size of beads?
These people have not, they have tried with a few different sizes of beads because they don't have very good control on the bead size. So, I mean, their beads have some distribution in size. So, that's all what they have tried. They have not tried very different bead sizes.
So, would you expect the curve to be different for different sizes of beads?
Yes, the curve will be different for different sizes of the beads, but the qualitative features will be the same. Okay. And also when you change the bead in this particular experiment, the force also changes because the size of the paramagnetic particle is different. So that also has to be taken into account. Okay. But the qualitative feature will be same unless you go to very small size smaller than the cytoskeletal mesh size, for example. In that case, the bead will do more like some kind of a, how do I say this? Some kind of a diffusion under a force. Okay, it will diffuse through the network holes. In one direction. Sorry?
Is it bounded diffusion?
Bounded
If the
diffusion.
size is more than the mesh size, cellular particle is more than the mesh size, then it should be some kind of a bounded diffusion.
Yeah, if it's more than the mesh size, then you can go to this kind of continuum kind of description of the of an elastic material through which, or viscoelastic material through which the cargo is moving. But if I go below the mesh size, then the particle doesn't really feel the elasticity of the mesh. The particle can just diffuse through the mesh. Okay? So like diffusion through a porous material, for example.
But what is the signature of the mesh that you see in this?
Oh, that okay.
It should not be like free some signature.
Yeah. So in that case, so you can, for example, you can look at the mean square displacement of the particle. Okay, that would be a measure which will tell you whether it's
Lower than the one.
Exactly.
But it will still be linear.
It will still be linear, but it will be lower than the free one. So those are. You can also look at the free diffusion. So you can also look at the free diffusion without a force. So then you will see that. Yeah.
So I am still confused. What in a material gives it this character? So is it, for example, the cytoskeletal elements give the spring
In this
portion and the axoplasm gives.
Okay, so in this particular case, it's the cytoskeletal filaments which is giving this property. The spring. Exactly, because when you push the bead through the material, you are stretching some of the cytoskeletal filaments. Okay, so if I take a network, if I take a mesh of cytoskeletal filaments, then this mesh has elastic properties because if you pull on it, I'm stretching this mesh, and then there is elastic energy stored in the mesh.
But what is the
So
lens?
in all viscoelastic materials, there would be two components. One, which would be the rigid component giving it the spring quality, and some liquid which would be providing.
Absolutely.
In what length scale this viscoelastic will convert into a diffusive bounded diffusion?
So I can only make a guess right now here. I would say that if the particle size goes below the mesh size of the network, then it will be more like a diffusion through a random media. And if the particle size is much larger than the mesh size, then it will see the viscoelastic response.
Think of the mesh size for actin to be about 50 nanometers.
In a cell, yeah. I don't know the value in axons. In axons, I would imagine that it's much denser.
Well, axon, the actin would be mostly in the cortex. The axonal axoplasm will not have any actin.
Yeah, so okay, maybe you know more about this. From the pictures which I have seen, electron micrographs, if you look at, you have the microtubule core, okay, and then you have neurofilaments outside, and then you have a cortical actin there. So there is no part of the axon which you can really say as just cytoplasm. There are filaments everywhere.
The central part would be microtubule, and microtubules are spaced. They are not
spaced, but cargo cannot interpenetrate, right?
I thought the space in between microtubule is generally
like proteins like tau. That's a kind of spacing.
Tau will be sort of decorating the microtubule.
Okay. Okay, so maybe then you must be more familiar with the detailed structure. So, for example, I have a picture here, which I can show you. This is the kind of picture I have in mind, where you have this microtubule array. They're cross-linked by protein like tau, for example, and also motors can also cross-link these microtubules. And so cargo, by and large, is restricted to the outer part, where it's full of neurofilaments and actin. This is not true, what you say?
But generally, I mean, that could be also a possibility. But the inner space with what you have drawn is actually, at least from the electron microscope picture, is quite large.
Quite large, okay. This the spacing between microtubules.
The space between the microtubule would be of the same order as the cortical thickness.
Okay. So suppose I say that these are microtubules which are spaced by a much larger amount. Then there are neurofilaments and actin here.
Spaced at least by 100 nanometers.
100 nanometers. Okay.
Yeah, 50 to 100.
Okay. So that's That's good to know. And but it's filled with neurofilaments then, right?
The neurofilaments are in the periphery.
Okay.
In the cortical region.
So there is something which restricts the neuro.
I think the thing probably depends on the time.
Yes.
Type depends on the
type of the axon.
type of axon. Okay.
Not all axons will be similar.
I tend to favor this outer thing that can move on the outside. But I think the larger diameter neurons that doesn't appear to be.
Okay, maybe because you see also if you look at electron micrographs, sometimes you see these scattered kind of microtubule present. Yeah.
Also, it will vary from insect to vertebrates.
Okay. Yeah. So one of the reasons why I am tempted to put this picture up is because when we look at transport in axons, it's not what we study, but we always see transport. Then you see that the membrane always gets sort of bulges out when these vesicles are going.
Absolutely.
And which gives the impression that they are in the periphery and not in not deep inside. But this may not be general. This may be only for
the axonal aperture is of the same order or bigger than the smaller than the organelle.
Then it has to bulge. Okay.
That's what happens in Drosophila when mitochondria pushes.
Okay.
What would you expect when here you are switching on the electromagnet and bead first requires and then stabilizes at a certain position? What would you expect if we start pulling it from behind? Means we switched on another electromagnet
in the opposite direction.
Yeah.
You will get, so if you switch this off and switch on another electromagnet in the opposite direction, then you get the same response, but the direction is reversed now. So you flip this curve, okay? Because strain now is in the opposite direction.
But you also have spring already. So if you start pulling that way, immediately then this will add up to that curve.
No, this won't, because here the bead, when I switch off and when the stresses on the bead is relaxed, it's as good as a freshly inserted bead.
Because it has to be some time for relaxation.
Exactly. So that is this time. This is the time for relaxation, and after this time, here it's as good as a freshly inserted bead. Okay. This is the time when all the stresses are relaxed in the system.
So I am asking, what will this behavior means during the relaxation, not after?
So during the relaxation, you get this kind of thing. In this, if you do if you do strain oscillations, if you keep moving it back and forth without giving it time to relax, then you get loops like this. If you put stress and strain, you get loops like this. Okay, because you are not given enough time for the stresses to relax. Even before the stresses are relaxed, you are moving into the opposite direction. Okay, so that is the reason why this curve goes up like that and comes down. I should have put some arrows here. It goes up like that, comes down, it takes a different path. And the reason is precisely that what you said. Is that okay or okay? Okay, so we are interested in mechanical responses and tension. So axon mechanics, you can broadly say that you can consider two types of mechanics. You can talk about active mechanics, which is related to growth and retraction of axons, and you can talk about passive mechanics, which is to do with stretching, sudden stretching. Okay. So one example will be like during embryogenesis, the axon is growing because the growth cone is extending, and at some point there is a synapse which will be established, and beyond that point, the axon continues to grow not because of the growth cone motion, but because of the tissue expansion. Okay, and if you so this will be growth, and retraction happens. For example, when you during path finding, often there are multiple synapses from the same axon, and not all synapses get established. So when a synapse fails to get established, then it is retracted back. Okay. So these processes are what I call as growth and retraction. Passive fast stretching has to do with, for example, if I just flex my arm, then the axon which is running through my arm may get stretched locally, okay, just because of the motion. Okay. And another place where this becomes important is stretch injuries. Okay, there are people in medicine who are interested in studying stretch injuries. What happens when you have sudden abnormal stretch? Okay. So why is axonal tension important? Here are just a few reasons. As I already mentioned, axonal growth and retraction. There is something called length minimization. Often what happens is during the pathfinding process, the path which the axon takes may be a convoluted path. It may not be the shortest path. But the job of the axon is to conduct signal. So you don't want unnecessary increase in length. So what some people hypothesize from observations in vivo is that once the synapse is established, the axon develops tension. Okay, and this tension helps the axon to become straighter. If you assume that the tissue is more like at very long times, like I talked about Mount Everest, like at short times the tissue is like an elastic object, but at long times the tissue can rearrange because cells can move around and so on. So the axon can, because of the tension in the axon, it can shorten. There is a recent experiment which shows that if you take an axon, maybe I should have put some pictures, I will just draw it here. So this is a growth cone which is growing towards a target and trying to establish a synapse. Okay. Now what these scientists did was they cut this axon. Okay. So when they cut it, this growth cone continues to move because it has some mitochondria and so on. For some time it can continue to move, but it fails to form a synapse. Okay. And then you might say, okay, I have cut it, so it's not surprising that it doesn't form a synapse. But if you cut it and if you take the loose end, let's say you grab it by your hand and you pull it, they do it with a needle, calibrated needle. If you pull on this loose end, then it forms a synapse correctly. Okay. So there is some indication that the tension in axon is important in synapse formation and normal functioning of the axon. There are also people who claim that the cortical folds which we have in human brain is forming because of the tension which is there in the axon. Okay? You can look up these references. This is controversial. I put two references. One supports this argument, one gives some arguments against it. But those who are interested can look up. I can also give it to you later if you don't have time to note down. Yeah.
Does that mean that in an organism, something like a Drosophila or a C. elegans, so synapse formation might be affected by the body size?
By the body size, I do not know. But there are experiments done in vivo on Drosophila, for example, where they open the Drosophila embryo and then they pull on the axon and they actually measure an active tension developing in the axon. Okay, there's a person called I think one of the authors in this reference, Saif, his name is I forgot the initials. So they do experiments in vivo. There's one more person in a different group. This person is illustrated. Okay, so I will describe some of the classic experiments which are done looking at response of axons to tension. Okay, so this experiment was done by Dennis Bray in 1984. The experiment is very simple. What he did was he took a glass needle, a very rigid glass needle, okay, and he coaxed the growth cone to climb onto the needle. Okay, he did that by coating the needle with laminin or some protein like that, which the axon likes to attach to. After the growth cone is attached to the needle, he pulled the needle with using a motor at a rate of about 1 micrometer per minute. And what he observed was that if you do this, then the axon length keeps increasing in response to this pull which you are applying without any change in diameter. Okay? What that means is that this length increase is happening due to addition of fresh material into the axon. Okay? So it's a tension induced growth of the axon. From this, he concluded that what the growth cone is doing is to generate a force, a pulling force on the axon, and this pulling force makes the axon grow. Okay? If you stop pulling, then the axon stops growing. Yeah.
You know, axons also grow after the synapse has been, that is, the two ends are anchored. When that happens, I mean, where is the membrane?
So after once the synapse has been established, there is it can still grow due to tissue expansion. Okay, if the surrounding tissue is growing.
Not only tissue expansion, no? You look at the height of an organism, like even humans. Certain synapses are formed early on, and the organism grows in length.
So here, one example I have given here some numbers is that if you take, I think this is for human motor neurons which goes into your leg. Okay, in the embryo, these neurons, the synapse is formed at if I remember correctly, 5th week or so. Okay, so the when the synapse is formed, the length of the neuron is only 1 centimeter. Okay, after that the growth cone stays put. It's not going anywhere. It's a synapse there. Yeah, there's a synapse there. But the tissue grows. Okay, during the growth of the person, the axon lengthens from 1 centimeter up to a meter. Okay, so this lengthening is coming because of the stresses exerted by the tissue on the axon.
So where is the membrane we have it?
So membrane is okay. So this is a good question. In this talk, I am not going to get into this, but there is a whole this thing about membrane addition. Okay, because you need, it's not just membrane. You also need to add microtubules. You need to add neurofilaments. You need to add actin. Okay, so whole material has to build up. Okay, so there is some mechanism by which the cell is sensing the mechanical stress which is being applied, and in response to that mechanical stress, it synthesizes fresh material. Okay. Where things are being added, maybe if I have time, I'll come to later on, because there are experiments people have done. For example, they decorate the axon with beads and see where the lengthening is happening.
Does the axon behave like an ideal material that it extends uniformly?
Next slide. Okay, I'll come to that in the next few slides. So here they are not probing any of those things. Here it's just a stiff needle. You just pull. It's like somebody dragging you with all the force they can apply. So the only thing an axon can do is to go with it. But the question you are asking is it stretching or is it growing? And the way they answer that is by looking at, is the diameter becoming less or is the diameter remaining constant? So what they find is that there is a volume increase. What's Krishna, how's time?
Well, we're doing fine. The lunch is already ready.
Okay.
So you can take another 15.
Okay. So now what happens if you do what you said? Okay, you apply a sudden pull. Okay, not a slow pull, but sudden pull. So that is what you see here. In this particular case, they have what they have used. So it's from Heidman's group. References below. Okay, I just change the way. You go with a glass needle. Okay, somewhere in the middle of the axon. So imagine the needle is coming from the top, and you pull on the axon. So this is glass needle. And this glass needle is a flexible needle. It's not a rigid needle like the earlier case. Okay, and it's a pre-calibrated needle. So by looking at how much the needle bends, you can calculate. And if I know what is the bending constant for this needle, then I know how much is the force. Okay, so I can calculate force. So that is the data which I have shown here. Is it clear how the experiment is done, or is there any question? Okay, so imagine this axon. The axon is like a guitar string. Okay, attached at two ends. The rest of it is free. I go with a needle and I pull. And my needle is bending because it's a flexible glass needle. And I know if the needle bends by 1 micrometer, I know what is the force needed to bend the needle by 1 micrometer. Okay, so from that,
how does the needle attach to the middle of the axon?
So they do in two ways. In this case, it's just mechanical contact. It's not attached. It's like plucking a guitar string. The other thing which they do is like what Bray did, which is to coax the growth cone to climb onto the needle, okay, by coating the needle with some laminin or some protein like that. Okay, if they do that, they see this kind of three-phase response. Okay, you are already familiar with this now. So they see an instantaneous stretching. Okay, then they see a viscoelastic relaxation, and at long times they see a slope, constant slope here. Okay, so then they do this experiment by applying different amount of force. So you apply 4 steps and change the amount of force which you are applying, and what they see is that if the force or the tension is less than 1 nanonewton, then the axon behaves like a viscoelastic solid. So you can model it by something like that. The spring is there now because of this instantaneous stretching. And if you apply a tension which is larger than, or force larger than 1 nanonewton, then it shows this liquid-like behavior at long times. Okay, which is then modeled by adding a viscous element here. Okay, so whether the axon behaves like a liquid-like viscoelastic liquid, or whether it behaves like a viscoelastic solid, depends on the force which you are applying. Okay, there's a threshold for this force. This, I think these experiments are done for PC12 neurites, so they're not real axons, but they're also done for chick embryo neurons, in which case this threshold value is higher, but still there is a threshold. Okay, what I have not put here is that what they also see is that if you reduce this force way below this threshold, then the axon begins to retract. Okay, so they also say that there is, depending on this, there are 2 thresholds. If the tension is very low, then the axon retracts. If the tension is in this intermediate value, then it's like a viscoelastic solid. At high tension, it keeps growing. Okay, what is called a tau growth.
Dissolve the active filament to this.
Sorry?
What happens when you dissolve the active filament?
Using latripilin or something like that, is it?
Or stabilize the active filament?
I am not sure if they have done force measurements. One of the problems is that if you attach the needle with the growth cone, then if you dissolve actin, then the growth cone just detaches. Even if it is not attached to the needle, this substrate adhesion, I think comes off if you depolymerize actin. I think this is a complication, if I remember correctly. So I have not seen data for that. But people like, I think Peter Bass will be here soon. So they have done experiments where you don't measure force, but you just disrupt actin and see what happens to retraction. And you see that the axonal retraction is inhibited if you remove actin or if you inhibit myosin with blebistatin. So I hope he will be talking about it because he has very interesting ideas of force balance inside the axon and all. So I am not getting into the microscopic mechanisms here. These are like more macroscopic measurements. What we are now trying to do is to actually address these questions with force measurements. So, okay, so these are experiments which we did some time back. So again, here this is the scheme of the experiment. You pull it with a glass needle. The glass needle is flexible. There's a motor which can displace this needle. And here are pictures. This is the unperturbed axon. The bright spot is the tip of the needle because light gets scattered from the tip of the needle. And then you displace it. Okay. In our experiment, we had a surprise. These are for PC12 neurites. Okay. Some axons behave like viscoelastic solids, and you can see, now you're familiar with these curves. Okay, so you can immediately say this is a viscoelastic solid. Some axons, instead of stabilizing at some constant value or keep on lengthening as we would expect for a viscoelastic liquid, some axons actually contract. Okay, that means the strain decreases. There is a negative strain rate, and this you don't get from any passive object. Okay, passive materials, if you take a rubber band or any viscoelastic material or a viscoelastic liquid, you pull on it, it will stretch. It will not shrink. Okay. So this has to come from activity. And what is active in the axon? One possibility is a molecular motors pulling on the cytoskeletal filaments. Okay. So that is what we did. So what we said was that, okay, now the passive axons, where you see this saturation, can be modeled as before by spring dashpot combination like this. But for an active axon, you have to also include a molecular motor element, okay, a new element. And what does it do? Molecular motors, maybe Roop mentioned this, or he can correct me if I am wrong. They have a characteristic force-velocity relation. Okay, what I plotted here is strain rate or velocity, and this is tension. Okay, so what happens is that the molecular motor are attaching to the filament and
no, sorry.
Okay, So the molecular motors are working on the filament, and if I apply a load or if I apply force on the motor, then the velocity changes. Okay, and the force velocity relation has some curve like this. That is, the maximum tension the motor can generate is at zero velocity or zero speed. Okay, and if the motor speed velocity is higher, then the tension keeps decreasing. Okay, so one way of understanding it, at least the way I understand it, maybe Roop has something to say about it, is that the motor is binding and making these conformational changes. If you pull the motor at a rate which is faster than the binding rate, then the motor is essentially unbound. You're not giving it time to bind, and then the motor is essentially slipping. Okay. So the maximum force the motor can generate is when you are not pulling on it. Is that a reasonable?
But in this case, you might be looking at the ensemble response of the entire structure.
Of course, yes.
And that, if at all, if any motor is involved, that would be myosin in this case.
So we are not assuming what motor is involved, because we have not done experiments where we probe. For example, we inhibit specific motors.
I would simply just concentrate on the actin and the neurofilament.
Yeah. So in these experiments, we have not done any inhibition of motors. Okay.
From here, the neurofilaments will be much more predominant compared to the
neurofilaments will be predominant, and there are also motor binding domains on neurofilaments. So in principle, one could also imagine, for example, a motor which is attached to neurofilament as cargo and attached to microtubule on the other side.
Those will be relatively, those forces will be relatively less frequent.
Probably, yeah. So,
I mean, their contribution will be much less compared to the contribution, the elastic force contribution of the neurofilament itself. It is the most stable and rigid.
What he's saying is this is contract.
exactly.
So because of the plasticity, you will not get a contract. You can't go the other way.
Exactly.
Right?
Exactly.
Yeah, that could be because of the active myosin
Yeah, but no passive mechanism will give you a negative slope. So that's what I'm saying.
So that, therefore, you think there is an active force.
Exactly.
The active thing is maybe motor, maybe something else.
Yeah. So where that active force is coming from, what is a microscopic mechanism, that we have not looked into. Okay? So that is what right now, that is what we are trying to address in the
One can probably add levistatin and see, inactivate the myosin.
for example, yeah.
And then inactivate kinesins and so on.
Exactly. The blebistatin is easy to perform, at least relatively easy. For microtubule motors, there are no well-known drugs available as far as I know. I think. Are there some? Huh?
I happen to
Like monostrol, for example, is only for a particular